Okay, now in this video lecture we will start to study the RNA-Seq experiments. And the RNA-Seq, again we will start from fastq files, then we will proceed with trimming, then we will do mapping, but now a bit differently than what we did with DNA-Seq. So we will use a tool called star for this, and we will explain the concept of the GTF file, and then we will see how to properly count how many reads map to each gene with feature counts, and for this we need also the strand of the experiment. And then to find which genes have been differentially expressed between two conditions, we'll proceed with HR. So to remind you the whole concept of RNA-Seq, we have a tissue and consists of many cells, so on. Then we extract the RNA from these cells. It's supposed to be some kind of homogeneous tissue here. We extract the RNA, and we have of course RNA from different genes. So we'll make some green RNA here, some blue RNA here. Perhaps this blue gene is expressed more, and some red over here. So let's say that's also a bit longer. So of course this is RNA, all of it, and it's also mature RNA, which means the splicing has already taken place here. Now so this consists of exons actually, not introns so much. So what we do next is that again we proceed with we fragment these RNAs, and then we choose the fragments of the right size. So here we have fragmentation. Size selection. As we do with all NGS experiments. And then we proceed with sequencing. So a few things that is worthwhile to remember are the following. These RNAs here may come only from one strand of the DNA. Why? Because expression may happen from one of the two strands of RNA, not both of them. The other thing that we need to remember is that we don't have introns here, which means that it's possible that a read, let's say that we take a read from this red one, we have this fragment, so this fragment, we proceed with sequencing. Let's say that we sequence this part and this part, paired-end sequencing. It's possible that this fragment actually, this read, sorry, comes from two distinct regions of the genome because of splicing. So it is possible that if this is the genome, and from here to here are the borders of the gene, let's say that this this fragment actually is somewhere here on this exon, and somewhere here on this exon. So what may have happened is that this fragment came from alternative splicing of, let's say, this and these regions of these two exons. So this is exon 1 and this exon 2. Because of alternatives, because of splicing and removing of introns, the RNA that was generated was this one. I just merged this piece together, and now this one is actually this corresponds to this and this piece. So this fragment here is actually this fragment here that comprises two different exons. And it's possible that this read here came from the end of the first exon and the beginning of the next exon. And therefore, if we try to map this back to the reference genome, it cannot be mapped precisely because some piece of this read comes from this exon and some piece of this read comes from this exon. And that's why we need a special software for mapping, and this is STAR that we will see later. This is splice aware mapping. Okay. Then what we will do if we have done the mapping, so let's say that we have now this reference genome. I'll just remind you here that the mapping we do on the reference genome, not on the exons, not on the RNA, but on, normal DNA reference genome. So when we do the mapping, we may have mapped all these reads, something like that. Let's say that this region is gene 1, and this region is gene 2. And then what we do is that we count how many reads correspond to gene 1 and how many reads correspond to gene 2, and we will end up with a table that looks like this. So let's say that these are 10 reads, and let's say that these are 20 reads, something like that, and so on. So we'll have on the rows the different genes, and here on the first column we will have how many reads we find from each gene. To do that, we will use this GTF file, which will provide information about the start and the end of each gene and the start and the end of each exon. Okay, let's go now to, so this is the theory behind it. What we will see on the way is how to find the strand of the experiment. This is important because these RNAs, this GTF file describes the structure of the genes, which means from which strand and where it starts and ends every exon and every gene, and from which strand it comes, the plus or the minus. And because on the genome there are overlapping genes, one for example from the plus strand and the other from the minus strand, but overlapping in the same region, it is necessary to consider the strand of the experiment in order to know from which strand, from which gene the RNA is derived. We will see that. Okay, let's start now. So for these videos, or for this lecture, we got data from ENA, the European Nucleotide Browser, and specifically an experiment with C. elegans. C. elegans is comfortable for such lectures because it's a small genome, doesn't require so much space, and actually it's a very interesting model organism as well. So here we have complete transcriptome of some knockout C. elegans, BIR2 knockout, and wild types also. With and without exposure to pathogenic bacteria Enterococcus faecalis. So we have two types of C. elegans genomes, wild type and knockdown of BIR2, and also two conditions with and without exposure to pathogens, so with and without pathogens. And we want to find out how the knockdown and the presence or the absence of the bacteria affects the expression level of the genes of the C. elegans. And if it affects, then which genes are affected. Okay, as usually we see here the experiments. And we can see also here, let's see, show column selection, experimental alias somewhere here. Okay, we'll go here to the bottom and move this bar to the right, and I see, for example, here on the sample title what every sample is. So here is the application 1 from a sample that is untreated and also wild type. So here we have a C. elegans without pathogen and wild type. Here replication 2 again, so 2 times, replication 2 again. So these are technical replicates, if I'm not mistaken. And then replication 3, replication 4, and 4. They have done many samples from every condition. And then they have BIR2 knockdown, so this is not wild type any longer, but also without a pathogen, untreated. They have done also BIR2 knockdown with pathogen, and so on. They have 49 total samples here. What I have done is that I have gotten the links of 12 of them, 6 of them. So if I'm not mistaken, let's see what I have already loaded in Galaxy. So I have loaded this 90, let's find it. I can go here to see what I have gotten the 90, 92, 95, 04, 05 and 09, 90, 92. So this is, here we must find the 90. This one is BIR2 knockdown untreated, so I guess that I have taken 94 also, 92, BIR2 knockdown untreated as well, and then 08, BIR2 knockdown pathogen. Okay, so if I'm not mistaken, I think that I have taken BIR2 knockdown 8, let's see the 9 what it is. BIR2 knockdown pathogen. Okay, great. So I have taken BIR2 knockdown all the time, either untreated or pathogen. So from all these experiments, I see the following setup. So we have a factor which is the genotype, and has 2 levels, the wild type and the BIR2 knockdown, and the factor that is pathogen, and again has 2 levels, presence and absence. So this is a two-factor experiment with two levels each, and the analysis requires some linear model actually. But because this is a bit advanced, what I did is that I got only the BIOL2 genotypes. I neglected those. So I got 6 samples. All of them are BIOL2. And then 3 of them have the presence of the pathogen and 3 of them have the absence of the pathogen. So I have an experiment that I can study how the presence of the pathogen affects the expression level of genes in BIOL2 knockdown genotypes. Okay. As usually, you can get the links from here. We see that it's a pertinent experiment, and load them in Galaxy, or alternatively you can get the TSV file, save it in your computer, and then extract the addresses, the URLs from there. We have done it many times in the past, so I will not show this again. Okay. I have loaded the data and I have put them, after I loaded them, on a collection. So this is a dataset collection, paired and dataset collection actually, as you see here. And now we can move to the workflow. I think that I have it here, yeah. Okay, so the workflow starts from is the following one. So this one is a list paired because it's a collection of paired end files. It's the 6 files that I described earlier. So this collection will be given to fastq. Fastq is a tool that performs trimming. Let's see what parameters I have put here. So there's a paired collection, the input. And then I don't disable adapter trimming, it's enabled. Disable no. Automatically it will detect the adapter, so I don't give anything here. I don't trim specific number of base pairs, so I don't put anything in the global trimming options because it's trimming how many bases in front of read 1 will be trimmed. I don't disable quality filtering, so if there are some bases that are unqualified, it means that if there are some bases in the read that are of low quality, and if there is 40% of the base pairs in a read of low quality, then the read will be discarded. It's the default, so I don't touch it. I can modify it, but it's fine as it is. And then here I do some automatic filtering. Again, it's the default values. But I cut also by quality in the tail 3' end, because the 3' end usually is of lower quality. So again, I do a sliding window similar to Trimomatic, 4 bases, 20 mean quality. So every 4 bases I check the quality, the mean quality of these 4 bases. If it is lower than 20, then I remove this window of 4 bases and I proceed. So that's all. We have done it in the past several times, and after that I give the output to Falco or to fastq in order to assess the quality of these reads. See, this is the first step, it's just a trimming step. Let's see if I have some results here of the Falco. So you see here the quality is actually quite good. 250 reads, 250 base pairs each read, quite long also. Something is happening here in the beginning. I'm not sure exactly what it is. It seems like some adapters have remained even though it's not be so. Some trimming has happened, that's why we see this distribution, but most of them are actually intact. You see that we have many reads with length 250, the whole length. That's all. Okay. So after we have done the trimming and we inspect the quality of the reads, we can proceed to the next workflow, which is the mapping. I have called this workflow C elegans star. So suppose that this input here, this one, is a trim data, and this is a star, and the star accepts 3 inputs besides the trim data that will be mapped. The first is the reference genome on which the mapping will take place, and as I said, the other input is the GTF file, which here is known as the gene model file, because it tells us where it starts, where it ends, every gene, where it starts and where it ends, every exon. We will see this file, and we can start with this file actually, yeah. So this file, I have it somewhere here. So is this GTF? I need the GTF, not the GFF3. I must have saved it somewhere here. Okay, so this is a file that consists of several columns. The first column is the name of the chromosome. Here we have the C elegans, that's why the name of the chromosome is not 1, 2, 3 like in humans, but it's V, for example, here. And then there are the second column is usually the name of the base, of the database from which they got this information here about the gene names and where it starts and where it ends every gene. So this is from the worm base. The third column is important because it informs us that this region that this line describes is a gene. So every line here describes, annotates, in other words, a region on the genome. And it tells us here that this region is on the chromosome V. It's a gene, and its coordinates are given here. So it's between this this position and this position. The next column is not very important, that's why there is a dot here. But the next one is whether this region, that's a gene, is on the plus or the minus strand, and here it's on the minus strand, as we see. And the next column, so here, again there is a region that has either dots or zeros, as far as I see here, I don't remember exactly what this is. Also not very important, but the important thing then is this one, because it gives us the name of the gene. And it's important to remember and to know what is exactly written here before the name of a gene. So here I see that it's gene underscore ID, and then there is a name of a gene. In other cases there is ID equals this one, and this is something that we need to remember. We'll need it later on. So for example, we see that from this position to this position there is a gene, and exactly in the same positions there is a transcript, which means that this gene makes one transcript. And this, between this position and this position, there is an exon. So this exon refers actually to this gene and this transcript here. And it tells us also here that the gene name that this section comes from is actually this one, which is the same as this one. So that's how we know that this exon is coming from this gene. And then here there are there is the actual coding sequence, CTS, again of the same gene, because this section might have some regions that are not translated. So the coding sequence, the position of the start codon, we see three base pairs here, 50, 51, 52. And then there is another exon, again of the same gene. Another CTS, another exon, another CTS, and so on and so forth. And then at some point we will go to the next gene. Here, it's the gene, you see the different name here. So this information is important. What we need to remember from here actually is this name here, gene ID. We will need it later on. And we need also to remember that since we have RNA-Seq, we need to pay attention to the exons. We need to get the name of the gene that the exon belongs to and focus on the exons, because we have RNA and the RNA comes from the exons. Okay, let's go back to Galaxy here, to the workflow, and let's see what we need to give here. Okay, so the input is paired-end as a collection. It actually refers to this box here. Here we will use the reference genome from the history we have downloaded already, and we'll create a temporary index. Of course, if we want to do mapping, we need to create an index. This one is rather important because it refers to some parameter which by default is 14, but it is related to the indexing step of the genome. We have said that before mapping we need to index the genome in order to make the mapping faster, and actually it's an obligatory step. It's not that if we will not put it then mapping will be slower. We need to index it. But since the genome of the C. elegans is rather small, the value 14 here will not work. The value 14 here is appropriate for humans, for example, or for some plants and so on. So for C. elegans we need to put a smaller value, and 12 is fine. And then we need this indexing that we will do to inform this indexing with the gene model, the GTF file that we already have. So here we see, okay, build index with gene model. Why this is necessary? Because we need to inform the mapper, the star program, where every exon ends and where every exon starts. So that's why we provide the gene model for this. Okay, then we will need to provide the GTF file that I just showed you before. And now we come to the important stuff that I told you earlier, what we need to know about this GTF file. So we want to use elements, reads, that come from exons. That's why here it asks us elements to use from the gene model, from the GTF file, to use for splice junctions. So the software star asks you how to understand where the splice junctions are. And we tell, okay, use the exon information from the GTF file, because where an exon ends and then the next exon starts, then maybe there we have a splicing. This we use by default 100. Normally we put the read length -1 here. So if we have 250, we'll put 249. But 100 in practice works pretty well. This is something that actually is not by default, the per gene transcript output, but I think it's quite useful to get. So star can already count the number of reads for every gene here. The default is nothing, if I remember well, no per gene or transcript output, but actually it's quite useful to have to get the gene counts from star as well. Deployed mode means if the reference that we'll give is a deployed genome or not. In our case, we have just a haploid genome as a FASTA sequence, so we'll say no deployed genome. We'll put yes here, then it will ask for a VCF file, which contains the information about the deployed genotype. So we have no here. We leave it no, the next one. We don't report chimeric alignments. This also, I think, by default, read alignment tags to include in the BAM output. You can have all these tags in the BAM output. This also by default 60, it's okay. Perhaps I have modified this, generated coverage for each strand coverage, yes. Maybe I want to see which every strand of the two, the plus and the minus, how many reads it contains, and that's it. Okay, so I close this. Then I have the RNA star. I say that it needs 3 input files. One is the trim data as a collection, the GTF file, and the reference file. This is a haploid genome, FASTA sequence. And then the output, so the mapped reads, it's a BAM file, will go to feature count software. And the feature counts here, it has a parameter. This software, the feature counts, will count how many reads every gene has. In order to do this, it needs a GTF file, again the same GTF file that we have loaded, so this one. You see here that it goes to 2 directions, one to the RNA star and the other one goes to the feature counts. Again, from the GTF file, what element we should consider the exon. And now it's the important thing that I told you before, the gene ID. So here we say that before the gene name, for example this gene name here, there is the flag gene ID. So when it maps something to an exon, it will know that the gene name that this exon comes from, it will know how to find it. Okay, so we need to open the GTF actually to see what's written in this column of the information before the gene name, and here is gene ID. The next one now is whether the input have paired or single end reads. It asks, does the input have read pairs? Yes, paired ends. And we have the option here to count the two reads of the same pair as two reads or one. So single end is one option, we don't have. Paired end, but still count them if individual reads. So two reads per pair, or paired end but count them as a single fragment, and I think statistically this is more correct. So here the two reads that come from the same fragment count as one. And the next we leave them as default and is fine. So here the important information that is not provided is here in the beginning, to specify the strand information. Indicate if the data is stranded and if strand specific read counting should be performed. Strand setting must be the same as the strand setting used to produce the mapped BAM input. This asks here whether the reads come from the same or from the opposite strand specified in the GTF file. This is usually specific for every experiment, but there is a tool in Galaxy that I will show later on that is helpful to find the strand. Usually for paired end, Illumina uses the stranded reverse, and for single end uses the stranded forward or normal, if I remember well. Here we have paired end and actually this correct stranded reverse. But it's something that you need to know what to put here. If you really can't find out what we have, then it's better to put unstranded. Okay. Okay, so this is a description of the workflow for the star, and if I could go back to the results of the Galaxy, then the output from the star will be the BAM file that we have seen several times actually. Let's see one. So this is the usual output from RNA star. It's a normal BAM file. Let's see if we can see it. That tells, for example, that this read with this binary, with this flag here, maps to chromosome 1 to this position. This is the cigar string, and this equals means that its mate maps to the same chromosome 2,671 bases away, and so on and so forth. So this is the mapping results, and then we have some other results also from star. It's these gene counts that we got for every gene. Let's see. Okay, here we have the gene name, and here we have how many reads have been counted on this gene, considering if the counts are unstranded, if we should take into account the first strand or the second strand. This is related to what I told you earlier, whether the reads come from the same strand as the GTF file shows for the gene. So for example, here you remember that we have we said that it's a reverse strand, is the second strand, and you see actually here that you see most of the reads actually are coming from the second strand also. So for example, yeah, this gene 1216 reads are coming from the second strand, only 5 from the first strand, and this is actually what we should consider. Now you may wonder why in the case of the unstranded case here this number is smaller than this number. Maybe somebody would expect that this plus this will give us this number, but this is not the case. And the reason is the following. So we have deployed, we have the two strands here, the plus and the minus. Let's say that the gene comes from this plus strand, and it's expressed this direction. Maybe there is another gene here on the minus strand that's like this. Let's express this direction. And there is this overlap here. So if some reads are coming from this region, and let's say that they come from this gene, so all of them are from the minus strand. So this minus means minus strand, this is just a read, so minus strand. If we say that this is unstranded, then the program will not know whether to assign it to this gene or to this gene, and therefore it rejects them. If we say, however, that this comes from the reverse strand, from the other strand, it means that if these reads will map here, if these reads will map here on this plus strand, then I will assign it to this gene that comes from here. This means that the reads will be mapped to the opposite strand of the gene that they are coming from. So this read comes from this negative strand, as I wrote here, but they will be mapped here. So I will assign it to the gene in the other strand. The same for this, the same for this, the same for this, and the same for this. So these 5 reads that would be mapped nowhere if I would put unstranded, because there is this overlap here, now they will be mapped to this gene. This is because I have this reverse strand protocol that tells me that if you have a read that maps somewhere on the genome, plus or the minus strand, then the gene that comes from is on the other strand. If I would have the forward strand protocol, then it would tell me that if a read maps to some strand, let's say this strand, then the gene is coming from a gene that is on the same strand, this one. If it maps here, then it comes from a gene on this strand. Okay. So this is it. I could actually get the read counts formation from here, but I use the feature counts, and in the feature counts I give the strandedness. So let's see what I get here. Now we'll see only one column. You see, I gave these numbers, large numbers here. So here I have only one column because I gave the strand already. It's considered to be the opposite strand. And these are the counts for every gene. So for example, this gene has 177 counts. Of course, I expect that longer genes will have more counts. Highly expressed genes will have more counts as well. Okay, that's for now, and the next video we'll proceed with the statistical analysis from several samples like that. So maybe I should mention here that I have one such file for every of the 6 experiments, for each of the 6 experiments. So here is another one, here is another one. The first 3 are from without the pathogen, and these 3 are with the pathogen. So what I will do in the next video is that I will compare the counts of these 3 guys with the counts of these 3 guys. Okay, that's it.